Welcome, fellow investigators, to this explainer. I am absolutely thrilled to have you here with us today. Let's just dive right in because today we are going to unpack the ultimate toolkit for figuring out how our world actually works. Now, whether you realize it or not, you and everyone else use the scientific method every single day. It's essentially an organized set of steps used to solve problems and answer questions about the world around us. So grab your virtual lab coats because we're going to master this process from start to finish.
I want you to think for a second. Have you ever made a weird observation in your backyard, or maybe noticed something strange happening in your kitchen, and you just felt that irresistible itch to investigate it? Maybe you wondered why one plant is growing way faster than another, or why your ice melts faster in one cup than the other one. Well, that curiosity, that is literally the beating heart of science. Every single great scientific discovery in history started with someone just looking around and asking, why?
All right, let's get into Section 1, Thinking Like a Scientist.
Before we can start mixing chemicals or building gadgets, we have to learn how to properly look at the world. And it is absolutely crucial to separate two things we actually confuse all the time: observations and inferences. In science, observing involves strictly using your five senses, what you touch, hear, taste, see, or smell, and you use those to collect factual information. It's just describing what is right in front of you. An inference, on the other hand, is a conclusion you draw based on those observations and your prior knowledge. For example, if you observe an elephant holding a peanut, well, that's a fact. But if you infer the elephant is going to eat the peanut, that's a conclusion. Actually, jumping to conclusions is what we want to avoid early on. As a scientist, you always want to start with pure observations before you make any inferences.
There is this magic moment of scientific inspiration. It's the moment where an everyday, normal observation transforms into a specific, testable question. And honestly, it's the single most important part of the scientific method. Every single part of your project is done to answer this one question. You go from saying, well, that's weird, to asking, under what conditions does this specific phenomenon happen?
Moving right along to Section 2, Forming Your Hypothesis.
Okay, so now that we have a solid, testable question, it's time to build our battle plan. We've done our background research, we've gathered our facts, and now we formulate a hypothesis. Now, you've probably heard a hypothesis called an educated guess, but honestly, it is so much more than a random shot in the dark. It's a highly structured prediction based on the research you've already done. And to make it perfectly testable, we use a very specific formula. Ready? It goes, if I change this one thing, then this specific result will happen because of this scientific reason. This formula tells the reader exactly what you believe will happen in your investigation.
Let's look at a super relatable 7th grade science fair example to see this in action. Let's say you're investigating plant growth. Your hypothesis would slide perfectly into our framework like this. If I use the most expensive fertilizer, then plants will grow taller because there are more nutrients in expensive fertilizers. Do you see how clean and testable that is? You know exactly what you're changing, what you're measuring, and why you think it's going to work.
That brings us to Section 3, Mastering Experimental Variables.
So this is arguably the trickiest part of the whole journey, designing the experiment itself. Let's say you wanted to test a different 7th grade mystery. Which brand of battery lasts longest in your gaming controller? To set up a fair test, you have to master your experimental variables. But don't worry, I promise you, by the end of this section, you're going to have a foolproof way to remember these for the rest of your life. Let's start with the independent variable. This is the one aspect of the experiment that you, the scientist, intentionally change or manipulate. In our battery example, it would be the brand of the battery you're testing. You should only ever have one independent variable at a time so you know for absolutely certain that the results you're seeing are caused by that one specific change.
If you ever get confused out in the field, just picture a wizard. Why a wizard? Well, because nothing in a magical storyline changes until a wizard casts a spell. It's the exact same with your experiment. You are the wizard. You have to start the change. The independent variable is the spell you cast to see what happens. Next up, we have the dependent variable. This is simply what you measure. It's the data that responds to your independent variable. If you change the battery brand, well, the dependent variable is how many hours the controller actually stays on. It literally depends on the change you made.
And your visual clue for this one, the ruler. Since a ruler is a measuring tool, let this visual help you remember that the dependent variable is all about identifying exactly what results you are going to measure and track. It's all that sweet data you write down in your logbook.
Finally, we have the control variables, which are sometimes called constants. These are the conditions that must remain exactly the same during your experiment. Think about it. If you're testing those batteries, you have to use the exact same type of controller, playing the exact same game at the exact same temperature. If you don't keep these conditions perfectly stable, there is no way you'll know if the battery brand caused the change or if something else entirely did.
So, for this one, think of control variables as the twins. Identical twins look exactly the same, right? Well, all of the control variables in your experiment need to look exactly the same too. They are strictly left alone so your test remains completely fair and scientifically accurate.
All right, let's test your new skills on a real-world scenario. A company wants to test a new diet dog food to see if it helps overweight dogs lose weight. 50 dogs get the new food, and 50 dogs stay on their normal diet. After a month, they check the dog's weight. So let's apply our tricks. The wizard, what did the scientists change? The diet dog food. Yep, that's our independent variable. The ruler, what are they measuring? The weight the dogs lose, that is the dependent variable. And the twins, that's our control group. The 50 dogs left alone on their normal diet, acting as a baseline for a perfectly fair comparison.
And finally, Section 4, Conclusions and Discoveries.
Okay, so you've asked the question, you've formed the hypothesis, you perfectly controlled your variables, and you ran the test. Now it is time to make sense of the data you've gathered. As you observe your experiment and jot notes in your logbook, you're actually going to be collecting two types of data. First is qualitative data. This is what you observe but can't measure precisely with numbers, things like shifting liquid colors or the shape and appearance of a reaction. Second is quantitative data. This is what you can measure precisely using standard units, like the exact temperature, the volume of a liquid, or a precise measurement in millimeters. And you know what? Both are absolutely vital for a solid conclusion.
Now, I need you to listen to me very carefully on this next point, because this is where a lot of young scientists get super discouraged. When you write your conclusion, you have to state whether your data supported or rejected your hypothesis. But listen, if your data rejects your hypothesis, that is not a failure. No way. In a lab report, disproving your educated guess is actually a huge scientific win. You just successfully discovered exactly what doesn't work. See, science isn't about protecting your ego or being right. It is all about uncovering the truth. A rejected hypothesis provides the world with vital empirical evidence. It literally tells future scientists what paths to avoid, which saves them a ton of time and resources. Every single result you get, whether it's exactly the one you predicted or a complete surprise, is a stepping stone. It guides you directly to your very next testable question.
And that brings us to the final step, communicating your results. Just like any enthusiastic, maybe slightly mad, but very organized scientist, you have to share what you found with the world. You do this by writing an abstract, which is just a short summary of your project, and a formal conclusion report where you explain exactly what your data tells you. Whether you present at a science fair, write a paper, or stand up in front of your class, sharing your empirical evidence is how the global community of scientists learns and grows together.
So, my fellow investigators, you now have the Scientist Toolkit. You know how to make keen observations, ask testable questions, formulate a highly structured hypothesis, and cast your wizard spells as independent variables. And remember, a failed hypothesis is just a new discovery in disguise. So my challenge to you is this. Keep your eyes wide open, stay infinitely curious, always look for the empirical evidence, and ask yourself, what mystery in your world will you investigate next? Thanks for joining me on this explainer, and happy experimenting.